All right, well, I'm going to go ahead and just get started with the intro as more folks joined in today. So welcome and thank you everyone for joining us today. My name is Lauren Moore. I'm the Chief Scientific Officer at the National Ataxia Foundation. And welcome today. Thank you for joining us for our science showcase, where we're going to hear about the characterization of naturally occurring mGlu1 mutations in SCA44 and SCAR13. So the work that will be presented today by Dr. Shane Hellyer is partially funded by a grant that an NAF grant that was given in 2021 for an early career investigator awardee. So I'm very excited to hear the progress that has been made on this program. So before we get started, just a couple of friendly reminders. So if you would like closed captioning, you're welcome to turn on closed captioning by using the CC icon from your Zoom menu. You also feel free to chat with other attendings using the chat feature in Zoom. However, if you want to submit questions to our speaker, please submit those using the Q&A feature. Questions will be answered at the end of the presentation, and they'll only be answered if they're submitted in the Q&A and not from the comments. During the Q&A session, you may also use Zoom's raise hand feature to request to be unmuted and ask your question live. And so with that, I'm excited to introduce our speaker today, Dr. Shane Hellyer. So Dr. Hellyer is a senior postdoctoral fellow in the endocrine and neuropharmacology laboratory at the Monash Institute of Pharmaceutical Sciences in Melbourne, Australia. In his doctoral and postdoctoral studies, Dr. Hellyer has established a track record in analytical molecular ion channel and GPCR pharmacology, including biased agonism and modulation of Class C GPCRs in recombinant cells and primary brain cell cultures. He's an emerging independent researcher with a burgeoning interest in naturally occurring mutations in G protein coupled receptors and how they contribute to the pathology of neurological disorders, especially spinocerebellar ataxias. So with that, I'm going to turn it over to Dr. Hellyer. The floor is yours.
Perfect. Thank you very much for that introduction. I'll just share my screen now. Good morning, good afternoon, everybody. Thank you for joining me today while I share some of the work that has been funded by a National Ataxia Foundation grant. So hopefully everyone can see my screen. I'll put on a laser pointer to make things easier. Yes, so as Lauren said, my name is Shane Hellyer. I'm a postdoctoral research fellow at MIPS, or the Monash Institute of Pharmaceutical Sciences here in Melbourne, Australia. I'm originally a Kiwi, which is why I have such a ridiculous accent, but I have been in Australia now for 8 years. And today I'm excited to share with you the work that I've done around some naturally occurring mutations in my receptor of interest, mGlu1, in relation to SCA44 and SCAR13. So I'm going to go maybe a little bit backwards and start with my acknowledgement screen, mostly because a lot of the work that you're going to be seeing today was carried out by some honors students that I've had over the last couple of years. So Ashwin Muralitharan and Leo Wang did a majority of this work, so I thought I would give them the credit at the start of the talk, and also to the rest of my lab, so my primary supervisor, Associate Professor Karen Gregory, Matt and Ash, PhD students around here that have also contributed to this work. And I wouldn't even be looking at ataxia without our collaborators, Russell and Shakib at the Children's Hospital, and their link to this will become apparent later as I give my introduction. And again, just really want to thank the National Ataxia Foundation for funding this research, and also to the National Health and Medical Research Council for the current funding that we have. So my area of interest lies in the glutamatergic system. So glutamate is the primary excitatory neurotransmitter in the mammalian CNS. So like most neurotransmitters, it is stored in presynaptic vesicles in neurons, where it is then released upon stimulation and interacts with receptors on a postsynaptic membrane to cause neurotransmission. That's a very basic overview of how basically all neurotransmission works, but I'm going to specifically focus on glutamate. So once glutamate has bound to its receptors, it has a number of different roles. So its main role is fast excitatory neurotransmission. So this means that your neurons are exciting each other and you're getting neurotransmission throughout various areas of the brain. It's also very important for synaptic plasticity. So this is weakening or strengthening of the synapse. So this area here, so how well your neurons talk to each other. And this is really important in learning, memory, and cognitive function, which I've got here. And for the sake of this talk, glutamate is really, really important in cerebellar development and function as we develop and also into adulthood. So if we look at these receptors here, there are multiple different kinds of receptors that neurotransmitters can bind to. The two main ones for glutamates are ionotropic receptors, so that's ion channels here, and metabotropic, so G protein coupled receptors. So why they are called G protein coupled receptors will become apparent in a minute. But basically, when glutamate binds to an ion channel, it opens, it lets ions through into the cell, you get depolarization, and this is a really fast response. So this is that fast excitatory neurotransmission that I was talking about before. This is a very kind of simple system. Your channel is open or it's closed, it's letting things in or it's not. You can have channels that run in the other direction, but for our purposes, this is a channel that lets sodium and calcium ions into the cell. So this is quite a fast response. And then you also have these kind of these other receptors. So metabotropic receptors act in a much slower manner. And upon glutamate binding, what happens is you have this G protein here. So this is why they're called G protein coupled receptors. So a G protein is a trimeric protein that is bound to the receptor. When you get agonist binding or glutamate binding, this dissociates into its parts, and then these go on into the cell to interact with other second messenger systems, which are numerous, and I'll talk about some of those in a minute, to kind of have more subtle effects on the cell, which can be things like slow excitation, gene expression, and things like that. So the mGlu receptors, or the metabotropic glutamate receptors, are my receptors of interest, and they fall broadly into three groups. So group 1, group 2, and group 3, and these are based on their pharmacology and their signal transduction pathways. And the group that I'm particularly interested in is group 1, which is made up of mGlu1 and mGlu5. So this is mGlu1. mGlu1 is one of the most important glutamatergic GPCRs in the CNS. And I'll just walk you through the structure of this protein. So the external part of the protein here, so if we imagine there's the cell membrane that lies here, so this is outside the cell, this is where the glutamate binds, and this is called the venous flytrap domain, and it's so called because it looks kind of like a venous flytrap. So it actually kind of closes around the glutamate molecule when it binds, and then binding of that glutamate molecule is transduced through the structure of the receptor through a cysteine-rich domain down to the 7 transmembrane domain, which is actually common to all GPCRs. So this part here is exclusive to mGlus and other class C GPCRs, but this part here is a classic kind of GPCR. And then we have a C-terminal tail here, which is just drawn because it's not well-defined in structures that we have. You can also see here that this exists, mGlu1 exists as a dimer. So if you draw a line through here, these are 2 separate mGlu1 receptors that are joined together. And so this is required for mGlu1 function. mGlu1 is ubiquitously expressed throughout the brain. So it's really highly expressed in areas like the cerebellum, the hippocampus, and the prefrontal cortex. And because of its high expression in these areas, then dysfunction in mGlu1 is associated with a number of different neurological disorders. So Spinocerebellar ataxia, which is of course what I'm going to be talking about today, but also cognitive difficulties, schizophrenia, and then also in other parts of the brain, it's been implicated in epilepsy, pain, anxiety, addiction, depression, Parkinson's, you name it, and there's probably some data out there supporting a role for mGlu1 in a neurological disorder. So if we look a little bit closer at what this looks like within a synapse, so here we have glutamate being released from the presynaptic vesicles. Here, our mGlu1 is sitting in the membrane, and it's transducing its signal through what's called a G-alpha-Q protein. So this is the G protein I was talking about before. Different kinds of G proteins couple to different signaling within the cell. So G-alpha-Q is linked through an enzyme called phospholipase C to calcium signaling. So calcium signaling and calcium homeostasis is really important within the brain. And it's really tightly regulated because you can get such functional outcomes as excitability, synaptic plasticity that I was talking about before, and protein synthesis. But you don't want too much calcium because calcium is also involved in things like cell death pathways. So if we kind of focus in on the cerebellum and the role of the cerebellum, the role of mGlu1 in the cerebellum, sorry. So mGlu1 is highly expressed in Purkinje neurons. So Purkinje neurons are the primary output of the cerebellum. And it has a really important role in cerebellar LTD. So this is long-term depression. This is a form of synaptic plasticity that I was mentioning before. So this is where the synapses within the cerebellum progressively become weaker. And while it may sound counterintuitive, that's actually a form of learning. So this is really important in our development in motor learning. And as you can imagine, if you have disruptions in this, then you have disruptions in motor learning and things like ataxia. It also plays a really important role in synaptic pruning during development. So if you see on these diagrams here, in utero and in your young life, your Purkinje neurons, which are here in green, actually are innervated by a lot of different climbing fibers, which are here in red. And as you grow and develop, and as the cellular functions change, these slowly get pruned off. And so as you can see, as you develop, eventually you are left with a single climbing fiber per Purkinje neuron. So this is a normal adult cerebellum. And this is brought about through partially through mGlu1 function. So you can imagine if you have disruptions in mGlu1 function, you can have disruptions to the way that your cerebellum is wired. And so as I mentioned before, calcium signaling within the brain, but especially within the cerebellum, is really tightly regulated. And mGlu1 is a really important partner in regulating calcium homeostasis within the cerebellum. And so disruptions in mGlu1 and also in various related calcium signaling pathways is linked to different ataxic syndromes, but specifically spinocerebellar ataxias. So I'm sure I'm preaching to the choir here a little bit when I talk about SCAs, but spinocerebellar ataxias are a group of rare genetic neurodegenerative disorders which are characterized by progressive loss of balance and coordination, cognitive impairment, cerebellar atrophy, slurred speech and sensory symptoms. These aren't always present in every single subtype. There are over 40 different subtypes, so you kind of get a mix and match of these. The two major groups are the repeat expansion mutation SCAs. So these are commonly polyglutamine repeats, and this results in kind of misfolded and aggregated proteins. And this is what we would consider the common spinocerebellar ataxias. So I've just stolen this this diagram here from a really good review. And you can see that if you have these extended polyglutamine sequences, you can get all sorts of different cellular deficits, and that's not going to be the focus of my talk. So if we move on to what I'll be talking about today, which are so-called conventional mutations. So these are missense mutations, deletions, duplications within genes that are related to various aspects of cellular function. And again, these tend to hit the Purkinje neurons quite hard. These are quite delicate neurons. And these mutations can be in important cellular mediators such as enzymes, cytoskeletal scaffolding proteins, ion channels, or for the purpose of the talk today, GPCRs. And so that's going to be my focus, especially on mGlu1. So mGlu1 signaling has actually been shown to be dysregulated in multiple different spinocerebellar ataxia subtypes. So if we look at the evidence from different animal models, we can actually see that even in these polyglutamine repeat ataxias, so SCA1, 2, and 3, you can get changes in both the expression, so how much mGlu1 you have, and also the function, so how well it works within these different ataxias. And it depends on what model you're using, how long that polyglutamine repeat is, and then also within Spinocerebellar ataxia 5, which is a conventional mutation ataxia with a cytoskeletal protein called beta spectrin. There's some evidence that mGlu1 is mislocalized and doesn't make it to the synapse properly. And not just mGlu1, so within these different types of spinocerebellar ataxia, we can have mutations in downstream mediators. So PKC, protein kinase C, is actually a really critical downstream regulator of mGlu1, as is the IP3 receptor, which you'll see in a minute, and also this regulator of G protein signaling. So there's a lot of evidence here that mGlu1 and its related proteins are quite critical in the pathology of these different spinocerebellar ataxias. So it's all very well and good to have animal models, but what about in the human contexts? So there's actually a few reports out there about mGlu1 autoantibodies and people that have really high levels of autoantibodies presenting with a paraneoplastic cerebellar ataxia, so an ataxic syndrome that's basically caused by your body fighting against the fact that you have mGlu1. And then, again, more specifically for this talk, there have been missense mutations and deletion mutations identified within mGlu1 that have since been labeled as SCA44 and SCAR13. So that's what I will be focusing on today. So as I just said, SCA44 and SCAR13 have been identified to be caused by naturally occurring mGlu1 mutations, and there's actually quite a few of them. So this is just kind of a map of where these mutations lie within the structure of the protein. And here is just the kind of the single code nomenclature for these mutations. So this is a tyrosine to a cysteine switch, leucine to a phenylalanine, and so on and so forth. And you can see they kind of cover the entire structure of mGlu1. And so SCA44 and SCAR13 are exceedingly rare. So these are kind of found in only a few families worldwide, a few individuals within these families. And you can see they are spread all around the world. And actually these two here, so this K981R and the P1068H, these were identified within pediatric patients in Australia. So this is the Sydney Children's Hospital collaborators that I mentioned to you before. So these aren't officially classified as SCA44 or SCA13 yet, but we're working on identifying how these fit in within the SCAR nomenclature. But this is actually, we got an email from Russell and Shakib about 4 or 5 years ago, asking for our expertise in looking at what the functional consequences of these mutations might be. So that's actually what kicked off my interest in ataxia and kind of what's led me here today. So I've included those here just as a bit of context, but I won't be talking about those because we're still working on that. So at the time that I got my Ataxia Foundation Early Investigator Awards, these were kind of the main mutations that we wanted to look at. So if we go back and look at some of these, so for example, this arginine 297 and this glycine 240, you can see that this asterisk here means that after this amino acid, the protein is truncated, so it's cut off. And so you can imagine that if you cut it off here, you only really have this part of the receptor. And so that's just a complete loss of receptor. There is no receptor to be made. And so we kind of excluded those from our analysis, even though we'd come across those in the literature for obvious reasons, because there wasn't really any point looking at those. So we became really interested in looking at these particular mutations. And you can see here that you can have some quite different symptomology with these subtypes. So the R in SCAR13 stands for recessive. SCAR44 is an autosomal dominant. This is recessive. So it's associated with kind of high levels of intellectual disability, a lot more in the way of the sensory symptoms, cerebellar atrophy, which is kind of a hallmark of a lot of these. And a lot of these recessive mutations are juvenile onset, so you get a lot earlier. Whereas the SCA44s, which is the dominant subtype, no cognitive impairment and quite late onset. And interestingly, this mutation here, so this frame shift mutation, actually you don't get any cerebellar atrophy, but you get some of the other symptomology, which is really interesting. And so you can actually see that here. So these are just some scans from the patients. And you can see here that in a lot of these, the patients have quite advanced cerebellar atrophy, but in this mutation, your cerebellum is kind of left untouched as far as that goes. So how these mutations affect the function of mGlu1 was kind of unknown at the time. There have been absolutely no work done on the SCA13 mutations and a little bit of work done on the SCA44 mutations. However, the original work used this kind of, so it's called a tango receptor construct. This is built to basically have a luciferase reporting outcome, which isn't part of the canonical signaling of mGlu1. And so it's not really an idea about the actual function of mGlu1 in relation to its calcium signaling, which is probably the most important part of its signaling. And what they showed was that actually these two tyrosine to cysteine mutations were actually gain of function. So if you look at wild type here in the black and the luciferase activity as a measure of activity, you actually get increased activity with these mutations and a decrease in activity with this truncating mutation. And then if you add in, so these are just two mGlu1 inhibitors, you can actually dampen down that overactivity. And if you add them both together, you get basically a return to the wild type. However, like I said, this is kind of a synthetic construct. It doesn't tell us a lot about the actual function of mGlu1 in relation to its signaling that we would see in the cerebellum. And also, this is kind of a single concentration of each of these drugs, and so it doesn't tell us a lot about the pharmacological manipulation. So when we talk about manipulating mGlu1 pharmacologically, there are kind of a couple ways we could do it. So glutamate, as I said, binds in this venous flytrap domain here. Because this is the recognition site for glutamate, you can imagine that this site is quite well shared across those 8 different mGlu subtypes and also on the ionotropic receptors because they're all recognizing the same thing. And so targeting this selectively in mGlu1 is really, really hard. So when your agonist binds, you get a response. We often use the analogy of the receptor as a light switch. So an agonist, for example, glutamate, is your on-off switch here. So you add glutamate, you turn it on. You add an antagonist, you turn it off. There are actually other sites within the 7 transmembrane domain of this receptor. So these are called allosteric sites, and these are where allosteric modulators bind. And so the real benefit of these sites is that these aren't as conserved between the different receptor subtypes. And so we can actually design molecules that specifically bind to mGlu1 and in theory will not bind to any of those other mGlu subtypes. And so that offers us the advantage of selectivity. And so what allosteric modulators do is quite cool, actually. They can directly activate the receptor, but what they mostly do is they act to, as the name implies, modulate the activity of your agonist here. So what they can do is they can modulate how well glutamate binds to the receptor, so that's affinity, and they can also modulate how well glutamate activates the receptor, so that's called efficacy modulation. So they basically act as, in the context of the light switch, as a dimmer switch. So you can basically turn the receptor activity up and down, not just straight off and on. So you have a little bit more subtlety in the way that you can change the spatiotemporal aspects of signaling. They also have a saturable effect, so they can't push glutamate beyond where glutamate can already go. So in theory, they're more safe. You wouldn't get as many cases of things like overdose, and they would be, as I mentioned before, more selective, so fewer off-target side effects. As I said, you can get some allosteric modulators that do have intrinsic agonism, which will come into play again a little bit later. So this is considered to be kind of a bad aspect of allosteric modulator pharmacology. So you can have modulators that enhance glutamate, or you can have modulators that inhibit glutamate, and these are called positive allosteric modulators, or PAMs, and negative allosteric modulators, or NAMs, respectively. So there's a lot of PAMs and NAMs out there for mGlu1, but there are a few limitations with existing drug discovery paradigms. So a lot of things will only look, a lot of discovery projects will only look at a single pathway. And if we look at, I don't expect you to remember this, but this is basically the signaling pathways for mGlu1. You can see that it activates a lot of intracellular pathways. And so looking at a single pathway doesn't tell us the whole story. And there is evidence for something called signaling bias, in which different modulators can activate different pathways. And so that's really important to look at that. We tend to use different agonists depending on what cells we're using. Sometimes we can't use glutamate, for example, if we're looking at neurons, because glutamate would activate every single glutamate receptor in there. So we use more selective synthetic agonists. And so this can lead to something called probe dependence, where we have different modulator pharmacology based on what agonists we're using. So that's a really important consideration. A lot of previous work had been done with rodent receptors. And so there is a little bit of difference between human and rodent mGlu1, especially within the binding sites for these modulators. So it's really important to know how those differences come into play. And also the way you design and analyze your experiments is really important for pulling out parameters that really tell you more about the pharmacology of these modulators. And so the broad aims of this project was originally to characterize the effects of mGlu1 allosteric modulators on human mGlu1 function. So this was really important because a lot of it had been done in rat, as I said. And then once we kind of had an idea about how these modulators were affecting the receptor, we were going to characterize the functional effects of the associated mutations, and then see if any changes in function could be restored by the allosteric modulators. So how do we test mGlu1? This is a much more simplified diagram of signaling. And the primary pathway that we use is calcium. So calcium, as I mentioned before, is one of the most important signaling pathways for mGlu1. And so what we do is we take recombinant cells, so HEK cells, human embryonic kidney cells, and we make them artificially express human mGlu1. We can grow these up in flasks, and then we plate them in glutamine-free media, so there's no glutamate floating around to confound our experiments. And then we can load them with a calcium-sensitive dye. So in the absence of calcium, this dye is quite dim. If we add an agonist that activates our receptor, we get influx of calcium from both extracellular and intracellular stores. The fluorescent dye lights up, and we can quantify that using a plate reader. So as our concentration of agonist increases, we get an increased fluorescent signal. Conversely, we can also look at IP3. So I mentioned before that the IP3 receptor was a really important mediator of signaling. So this is kind of the opposite, in which at baseline conditions, we actually have these two antibodies that interact with each other and form a fluorescent signal. So at baseline, we have quite a high fluorescent signal. When we activate the receptor and we produce IP3, IP3 gets broken down into IP1, and then IP1 actually outcompetes one of these antibodies, and we get a reduction in signal. And so as our concentration of agonist increases, we get a decrease in signal. And so we can go on and quantify these. So if we run, essentially we run a baseline first to see what the baseline level of fluorescence is in the cell, and then we will add either a vehicle or agonist, and then we can quantify how big the response is, either by the peak of the response or the area under the curve, depending on how we want to do it. And so then we can add increasing concentrations of our agonist, and we can quantify this as we go up. And eventually we have what's called a concentration response curve here. So this is a function of how much calcium in the cell versus how much agonist we're putting onto it. And so if we add a positive allosteric modulator, we can enhance this signal. And that is borne out in the concentration response curve here, where you see an increase in the calcium. And conversely, if we have a NAM, this flattens it out. And so we get a decrease in signal. And so from there, we can use non-linear regression analysis to pull out some parameters from this. So the two most important parameters for agonists are potency and Emax. And basically, these are how good is our agonist at activating mGlu1, and how much mGlu1 is activated. So that's the really simple way of putting potency and Emax. So these will be parameters that I'll talk about going forward. And when it comes to allosteric modulators, we again can use regression analysis. and we pull out 3 important parameters. So affinity, which is essentially how strongly does the modulator bind to mGlu1. So this is a comparison of the different modulators and how well they bind. And then log alpha and log beta are our what's called cooperativity parameters. So what is the modulator doing to the agonist? Is it positive? Is it neutral, aka doing nothing? Or is it negative? Is it inhibiting the receptor? And so the first part I'll go over relatively quickly. It's just some pharmacology of these modulators. And so kind of showing the importance of those limitations in the drug discovery paradigms that I was showing before. So you can see here that this compound, the J&J compound here, is a negative allosteric modulator of glutamate. So our glutamate curve is here in the open circles. And as we add increasing concentrations of J&J, we get a decrease in signal when we have glutamate as our agonist. So decrease in signal, we can pull out an affinity value and also these log alpha and log beta values. If we use quizqualate, which is a common synthetic agonist, we can see that this profile is essentially exactly the same. So you get a similar affinity value, neutral affinity modulation, and it's a full negative allosteric modulator with regards to efficacy. So that's no probe dependence. So probe dependence is if we had differences here. So CpCCOET here, which is a completely structurally independent NAM, you can see here that we're actually getting a shift in the potency of glutamate and also a decrease. And so whereas J&J was neutral with regards to affinity modulation, we're actually getting a difference here. And then if you look at CpCCOET modulation of quizqualate, it's going straight down. So that's essentially what we saw with J&J, so it's neutral. And what we have here is we actually have a different affinity parameter, a different log alpha parameter. And so we can say that CpCCOET is a probe-dependent NAM. And this becomes really important if I were to do experiments in a neuronal cell culture, where I would be using, for example, quizqualate instead of glutamate. And I'd have to kind of take this into account when I'm analyzing my data and interpreting that data. Speaking of different functional outcomes, as I mentioned before. So again, calcium assays, this is a PAM this time. So CpPHA is enhancing. So you see it's shifting to the left and up a little bit. So you're actually enhancing the glutamate curve here and also enhancing the quizqualate curve here. So with increasing concentrations of CpPHA, you're getting an increase in potency of these agonists. Again, probe dependent, but the real interesting thing here is if we look at a different functional outcomes, these are both calcium assays. If we then look at modulation of quizqualate in an IP1 assay, you can actually see that at baseline here, so this here is a vehicle, so there is no agonist present at all. This is just CpPHA. It's actually having its own agonism. So it's activating the system in the absence of the agonist, which, as I mentioned before, is potentially not a beneficial part of allosteric modulation. And so you can see that there's quite big differences between different functional outcomes and different agonists. So this compound is interesting. This Roche compound here was originally reported to not activate human mGlu1. And as you can see from our data here, we showed that it's pretty good at modulating human mGlu1. And if we look at the old data, and I apologize because this is the best quality image I could find from this old paper, what they actually did was they just tested a single concentration against a single agonist concentration, and there were no control curves or anything. So it's this kind of shows the importance of how you design your experiments and what conclusions you are drawing from those experiments. So based on this, they said that this particular compound had no activity at the human receptor, which if you kind of design your experiments properly and do this kind of more robust analysis, you can see that it actually does. So nitazoxanide is a really interesting one. So going back to the paper before that I showed you where they were modulating those gain of function mutations, they actually used this molecule called nitazoxanide, and they showed that it also decreased the activity of these gain of function receptors. So nitazoxanide is actually an FDA-approved drug. It's an antiviral, antihelminthic drug, and it was identified as an mGlu1 NAM from some in silico docking studies. And so what was exciting about this is the potential for drug repurposing. So you don't have to go through the same clinical trials and safety if your drug is already approved. And so we wanted to test this and kind of pull out some different parameters for nitazoxanide as a NAM. And when we put it into our system, we can see here that with increasing concentrations of nitazoxanide, you are actually decreasing the response to both glutamate and quisqualate, so both of our agonists. So it does look like it's acting as a NAM, which we were really excited about. However, it also acts as a NAM when you are using ATP. So this is not even acting on the glutamatergic system. This is a purinergic receptor agonist, and also carbachol, which is a muscarinic agonist. And so what's happening here, we think, is that it's having non-selective effects on the calcium signaling just within the cell in general. And I actually found some old data from a paper that showed if you add nitazoxanide here, you basically are increasing the calcium in the system. And so what we're seeing here is potentially system rundown. So you're actually overactivating the system, which then conversely means that you don't have enough calcium to then mobilize with your other agonist. So it's kind of this weird overactivation leading to inhibition of the system. And so in summary for this first part is that structurally distinct allosteric modulators have diverse pharmacology. And so we have some that have the intrinsic agonism that you don't see from our calcium pathway, which is our primary screening pathway. So this shows the importance of using different outcomes. The choice of agonist is very important based on the probe dependence that we saw. And also, unfortunately, the putative NAM nitazoxanide that could have been used for repurposing is actually non-selective. And so that would cause troubles if you tried to use that as a drug. However, the pharmacology of these compounds can actually help to inform us in treating mutant mGlu1 in Spinocerebellar ataxia, which is where part 2 comes in. So this is where we are characterizing the effects of our mutations on receptor expression and function and seeing if we can change this function back with allosteric modulators. So in this in this study, we used a tagged receptor. So we created a receptor that has the human mGlu1 with what's called a c-Myc tag on it. So we can use the c-Myc tag actually with anti-c-Myc antibodies that are fluorescent, and we can determine how much mGlu1 we have on the cell surface. So that's where it belongs. mGlu1 signals from the cell surface. And so if we quantify how much mGlu1 we have, so here's wild type, and here are those different SCA-associated mutations that I mentioned before. You can actually see that most of them, there's a little bit of variation, but most of them actually maintain their expression on the surface. So whatever's happening to their function, it's not because there's less mGlu1 there. It's obviously something to do with how the mGlu1 is working. That is with the exception of this G1056. So this is the frame shift mutation that I mentioned before with the that doesn't have cerebellar atrophy. And this is, so you're getting basically no mGlu1 on the surface of this for this particular mutation. And if we look at where this mutation is, we can actually kind of explain that away a little bit. And that this mutation here actually cuts off this entire part of the receptor. So this is the C-terminal tail. It actually cuts off this part of the receptor here and has this little truncated version of the C-terminal tail on it. And the important part about this part of the C-tail is that it actually contains a binding domain for something called HOMA. So HOMA is a really important scaffolding protein that actually helps mGlu1 get to the cell surface. And so if you don't have this recognition domain, HOMA can't bind to mGlu1, and so your mGlu1 will not make it to the cell surface. So that's kind of a nice explanation for potentially why we're seeing that decrease in this particular mutation's expression. So for these, for L454 and N885, we've showed that it's still there, but is it working as well as the wild type receptor? So first of all, we'll use our calcium assay, and you can see here that if you use glutamate as our agonist, wild type is here in the black circles and here in the dotted line, we can actually see that this L454F and N885 delete mutation are having different effects on glutamate versus quisqualate. But the take-home message is that here, they're causing a shift in potency. So that's how well glutamate is able to activate the receptor. Whereas with quisqualate, the N885 delete mutation doesn't have any effect on how well quisqualate is activating the receptor. It's basically just dampening down how much of the receptor is activated. So it's interesting to see these differences between the two different agonists. And if you then look at the IP3 pathway, you see a similar kind of trend where this L454F mutation is consistently shifting the potency of these agonists, and you're getting a decrease in Emax with this other mutation. And so this G1056 mutation is causing a complete loss of function. There's no response in any of our assays, and that kind of makes sense if you don't have any of it there on the surface. We included it just to make sure that we weren't seeing any activity at all. And then L454F is more of a loss of potency, and NAF5 delete has a mix of potency and maximum effect loss of function. And so take-home message here, don't worry about the numbers and the letters and things, is that these different mutations affect potency and maximum response in different ways. So you're getting this kind of mosaic effect. And what we can actually do is we can look at potency as a measure, because potency is actually made up of two separate submeasures. So potency is essentially how well does glutamate bind, and then also how well after it's bound does it activate mGlu1. And we can use our system to quantify this. So we actually have a tetracycline inducible system here, which means that until we add tetracycline, we don't get any receptor expression. When we add tetracycline, we induce expression, and we can actually titrate how much receptor we have using different levels of tetracycline. And then we can see what these different levels of receptor look like with regards to activation by glutamate. And again, we can then use nonlinear regression to actually pull out these affinity and efficacy values. So we can pull apart potency and see, are these mutations affecting affinity or are they affecting efficacy? And so what we can see here is that N885 delete is mostly affecting efficacy, so how well the glutamate activates the receptor, whereas L454F is actually reducing how well glutamate binds to the receptor. And if we look at where they are on the receptor, that kind of makes sense. So L454F is sitting right here. So this is in the glutamate binding pocket. So if you have a mutation here, then it's likely that you would change the recognition of this pocket for glutamate, and you would have lower ability to bind. Whereas N885 delete is sitting down here. So this is the once you've activated the receptor, you've transduced the signal down through to this part, which is then activating signaling within the cell. So again, it kind of makes sense based on where these mutations are and what they're doing, how they're affecting function. I'll skip past this. So glutamate activity was reduced at both these mutations. So you can see here that this is the L454F response in relation to the wild type response here in the dotted line. So we've got this massive shift in potency. Glutamate isn't as good as activating the receptor. Same with N885 delete. So what we can do is we can add in these PAMs, so positive allosteric modulators, at a single concentration and see what they do to this response. And encouragingly, what happens is that if you have glutamate in the presence of a PAM, you then shift this potency of the glutamate response curve back to overlay with the wild type. So essentially it takes glutamate plus a PAM, so glutamate plus a positive modulator, to be as effective as glutamate within the wild type, but you can actually get the glutamate signaling back to overlay with the wild type signaling, which is really cool. So that is for both of these receptors, and you're also getting this quite large increase in your maximum effect. So that's the loss of function mutations. For the gain of function mutations, we were a little bit disappointed when we first looked at our calcium signaling to see that this potential gain of function mutation here, so Y792, it didn't do anything. It overlaid exactly with the wild type. And so we're a little bit confused about that. And then we were even more confused when we looked at the IP3 signaling. So the IP3 signaling showed that this Y792 actually massively decreased the mGlu1 response. And so we were very confused about this. And we went forward with testing different modulators, and we actually found out that this was because this Y792 mutation has higher levels of constitutive activity. So that's mGlu1 activity by itself. So mGlu1 and its related receptors are known to kind of just tick over signaling in the absence of agonist, and so that's called constitutive activity. And what we saw is if we look at the wild type, you can get this beautiful IP1 signaling with our agonist, and we have some NAMs here, so these are negative allosteric modulators, and if you add those in, you get a small but subtle decrease from this baseline activity. So this is telling us that we have some activity at baseline that we can reduce with a NAM. And if we look at Y792C, we get a much larger decrease with these NAMs. So what that's telling us is that the activity at baseline of mGlu1 is much larger with this particular mutation. And so this mutation isn't affecting the ability of these NAMs to bind. They have similar potency values, but it is affecting their ability to reduce this baseline activity. And if we look at the baseline levels of IP1, so if we just let the cells sit without any agonist, so just add vehicle, you can actually see that Y792 does actually have higher levels of baseline. So if we just don't stimulate these cells at all, this cells containing this mutant receptor will produce more IP1 at baseline than wild type. So that kind of fits in with our hypothesis that there is some sort of constitutive activity, which is causing, again, kind of similar to the nitose oxide story, causing some sort of system rundown that means you can't activate it as well with your agonist. And so if we can also add in these NAMs in the presence of our agonist just to see how well they're inhibiting, and we can actually see that these NAMs are better, or one of the NAMs is better at inhibiting this receptor than it is the wild type. So that's also kind of a hint that maybe we're getting overactivation that can be dampened by these NAMs. So Y262 is the other gain of function mutation. And again, we didn't see anything in calcium. So we started to look in IP1 and started to look at these NAMs. And what you can see here is that you're not really getting much of an increase in the window like we did with Y792. But if we quantify the potency, so how tightly these NAMs are binding to the receptor, they're actually better at binding to and inhibiting the receptor. There may not be as much constitutive activity, so you don't get that increase in the window, but these NAMs are actually more potent at inhibiting the receptor, which is another hint that there is some sort of more subtle gain of function here. And if you look at them in the context of being there with an agonist, you actually get no difference. So it's something to do with this baseline activity that we can inhibit with these NAMs. And so if we look at where these mutations are and try and think about what they could be doing, it starts to make a little bit more sense. So this is an inactive structure of the mGlu1 dimer. Upon activation, you get this massive rearrangement and this tightening and rotation of the dimer. And if we look at where our mutations sit in relation to this, it actually starts to make more sense. Again, L454F sits right in this binding pocket, so that's probably why it's reducing the ability of glutamate to bind. But if we look at Y792, which is down here, in the inactive structure, they sit quite far apart. But then once you activate the receptor, which is here in orange, you get this massive rearrangement. They move closer to each other, and they're now in quite close proximity. So if you replace these tyrosine residues with cysteine residues, so cysteine residues can actually form disulfide bonds between each other. So it might be that if you have two cysteines here, you're actually holding the receptor in this active state, which would explain why we're getting that increase in constitutive activity. So you're kind of enhancing the receptor activity by locking it into the state that usually is only achieved with the agonist there. And again, with Y292, it's a little bit more subtle, but again, upon activation, they kind of move closer together. And so if you replace these with cysteines, then you might get some effects on the ability of the receptor to shift between these inactive and active states. So in summary, I hope that I've convinced you that we have some quite different effects of these SCA-associated mutations on the function of mGlu1. And so we have this L454F mutation that sits up in the venous flytrap domain that decreases agonist affinity. We have one in the 7-transmembrane domain that increases constitutive activity. And then we have some down in the C-tail that decrease the efficacy of the agonist, and also one that just completely kills surface expression and function. And so what was really exciting and what we had kind of set out to prove was that we could actually rescue the function of these mutant receptors using our allosteric modulators. So those with decreased activity could be rescued by those positive allosteric modulators, and the constitutive activity that we saw with Y792 could actually be reversed by our negative allosteric modulators. So this was a really cool kind of proof of concept, and this is what we'd set out to show. And so this comes with a few caveats, of course, and this is where our future directions come in. So as I mentioned, mGlu1 is a dimer. It dimerizes with itself. It can also dimerize with other members of the mGlu family. And where the dimer really becomes important is for those autosomal dominant SCAs. So you can imagine that in a dominant heritability syndrome like that, you have one mutant allele and you have one wild type allele. So there is the potential that you would have half of this receptor being wild type and half of this receptor having a mutation in it. And so we have no idea what that means with regards to function. Does that rescue the receptor? Does it make it worse? And so it would be really cool to be able to look at different dimer makeups. So all of the work I showed you today was in a recombinant cell line. So that's a very simplified cell line. And we would love to be able to recapitulate this in either neuronal cultures from mice, which we do quite regularly, or patient-derived stem cells, which is kind of gaining traction now as a really cool tool for studying things like this. And also, what is the role of mGlu1 dysfunction in those other forms of SCA? So as I mentioned in my introduction, mGlu1 dysfunction is associated with these polyglutamine repeat disorders. And there's a little bit of evidence out there now that you can actually activate mGlu1 to enhance clearance of some of these proteins. mGlu5, which is really closely related to mGlu1, has some interesting expression patterns. When you start to reduce mGlu1, you get these compensatory increases in mGlu5. So we're looking at how the group 1 metabotropic glutamate receptors are involved in these other forms of SCA. So I've got some current proposals in for funding for that. If you want to read any further around what I've talked about today, so we actually have a publication in Molecular Pharmacology. So that was Ashwin's work for the pharmacology. And then we have a preprint currently for the mutation work, which we have submitted to a journal. We're just waiting on reviews to get back to us for that. So thank you for your attention. Hopefully I haven't bored you too much with some pretty hardcore molecular pharmacology, and I'm happy to take any questions.
Thank you so much, Dr. Hellyer, for that presentation. And just a reminder to folks on the call, you're welcome to present a question to our speaker, either through putting it into the Q&A in the bottom of your Zoom, or by raising your hand and question directly. So I, of course, from a patient organization, I'm curious how these findings might lead to new treatments for the SCAs. And in particular, if there are treatments, when those treatments might be effective in the time course of ataxias, given that you showed some dichotomy between increased and decreased levels of mGlu1.
Yeah, so that's always an interesting question, and it's something that I kind of struggle with a little bit as a pharmacologist, because as you kind of alluded to there, in the context of a neurodegenerative disorder, a lot of the times you don't start getting symptoms until you have some pretty bad neurodegeneration already. And so being able to slow or reverse that is really dependent on when you're coming in with your intervention. And especially when it comes to genetic disorders, it becomes a question of, is pharmacological intervention the right thing, or is that, especially with the advent of things like gene therapy and things like that. So this is something that I think about a lot, and I think for the recessive ataxias, it will be a little bit harder, because you do get that juvenile onset, so you don't really have any lead time to kind of, to see it start to develop. It's kind of bang, it's done. And so I think the probably the most promising results from ours were more related to the SCA44 mutations. So if you could identify those early enough with genetic screening before you get that symptomology, and you can kind of dampen down that overactive mGlu1 with NAMs. I think that might be a viable approach. There are no mGlu1 NAMs currently in the clinic. mGlu5, which is the most closely related mGlu receptor, there's a lot of mGlu5 NAMs that have made it to even through phase 3 clinical trials for various disorders. So there is proof of concept there that these molecules are safe, are effective, and can have the kind of properties that you would want from a drug. So again, it's a bit of a hard question, but I think for the SCA44, that might be kind of where we'd be leaning with that.
I was also really intrigued by the lack of cerebellar atrophy, despite clear cerebellar ataxia. Can you explain or provide some insight into how that can occur?
Yes, so I kind of alluded to that a little bit at the end, in that I think for the majority of these mutations, you're getting these kind of, I guess, subtle but insidious changes, losses in gain of function in the receptor that over the course of time will eventually lead to disrupted calcium signaling and cerebellar atrophy. I think with that truncated, the G1056 mutation is that because it's basically you have no receptor right from the start, what can happen is that mGlu5 again, so mGlu5 is kind of my other baby receptor, I know a lot about that, is that if your neurons detect that you don't have any mGlu1 at all, it actually upregulates mGlu5, which can play a really similar role to mGlu1 in maintaining cerebellar health. So I think, again, this would be really cool to look at in like patient neurons or something like that. I think what is happening is that because you don't have any mGlu1, your mGlu5 is being upregulated and basically playing that role. That's kind of my hypothesis as to why you don't get any atrophy at all, because otherwise it doesn't make an awful lot of sense.
In that regard, do you think perhaps some of the cerebellar ataxia symptoms might be reversible if there is a sufficient presence of mGlu1 or 5, as you mentioned?
Yep. So that's kind of again where we're going with our more recent proposals, as we really want to dig deeper into how these levels of mGlu1 and 5 in relation to each other, and how they regulate each other, because they can also dimerize together, so that makes it even more complex. But yeah, I think if you can identify at any given point of the development of the ataxia what levels you've got of each receptor, then you can start to look at maybe we need to bump one up, bump one down, and then that becomes kind of more of a co-therapy type situation.
And before we close, I'm going to ask a more general question, which is, do you have any advice you'd give to a PhD student or early postdoc who is considering a career in ataxia research?
Do it, I guess. Like I said, mine kind of came around very kind of serendipitously from that contact that I had with the researchers from the hospital. But I think having the National Ataxia Foundation there is really handy. Not every, not everyone has access to those kind of small pots of funding that help you kind of get your research off the ground. So the National Ataxia Foundation, Ataxia UK, there's lots of them out there. And I think that's been really helpful for me, not only being able to kind of get really, I don't know, lots of good ataxia research all in the same place, like these kind of symposiums, all of the conferences and things like that. But also to get those little bits of funding that provide proof of concept that then look great on your CV to then go for the bigger funding. I'm really excited to continue looking at ataxias if I can. It all depends on who wants to give me money at any given time. But yeah, I'd say go for it. I think it's really fascinating. And I've also learning a lot about, engaging with people with lived experience. I know that people living with ataxia are really engaged with the research community, and I find that really awesome.
All right, well, we are at the end of the hour now, so thank you again, Dr. Hellyer, for your presentation, and on behalf of patients, thank you for all of the work and dedication you put into these ultra-rare disorders.
Thank you very much.
All right, thank you. Bye.